Yeah, so I'm going to tell you a bit about delayed array, and my pitch for it is that it is like a tibble for arrays. So I'm going to be mostly talking about work by other people, in particular Hervé Pages from the Bioconductor core team. So you might be wondering why I'm here. And that's because I've been an early adopter of this package and the framework. I've been using it in practice for the last year and a half or so to analyse large genomics datasets that otherwise would not have been feasible to analyse. And as part of this work, I've developed, I've adapted two existing packages for analysing DNA methylation data from the lab I was postdocing in, and developed a companion package to DelayedArray that use and extend upon this framework. So I'm here to tell you about my experience and to kind of do a bit of evangelising for where it might be useful in your own research. So to take a step back, one of the core data structures in Bioconductor is this summarised experiment. You can think of it as basically like a matrix with some annotations, and what it looks like is basically you have rows which describe some sort of features. So these might be genes, it might be SNPs, it might be other loci in the genome. And then the columns represent the samples. And for each of the rows, which are the features, and the columns, which are the samples, you have metadata tables that describe information about those. You might have more than one assay. So the assay is the actual thing you measure in your experiment. You might have multiple assays, which is common, for instance, in DNA methylation. And then you might just have some top-level metadata about the experiment, like who ran it, how many batches there were, things like that. What I'm going to talk about today is the assay data, so the actual measurements. And typically, these are just stored as an ordinary R array, or a matrix if you've got a two-dimensional version. So why is this sort of the standard data structure? Well, it does have some structure. It's not tidy in the sense of like the tidyverse, but it has a consistent sort of API where rows are features, columns are samples, and that's being shared and is like a familiar interface. And it's familiar to most R users. An array is a core data structure in R. An array has many packages that can make use of this. One that I particularly love is Matrix Stats by Henrik Bengtsson, which has a very nice API for doing column and row summaries of the data, so computing column medians or row variances. It's an array, so it's ready for matrix algebra. You often have to do those sort of operations in any sort of analysis. Tight integration to BLAS and LabPack, ready for C and C++ integration, and I think most importantly for genomics analysis and Bioconductor is it's very conducive to interactive data analysis. And exploratory work is it's a large part of doing high-throughput biology, statistical analysis. The issue that we're facing, though, is that modern datasets are often too large to be stored in memory as ordinary R arrays. I'm going to give you two examples of this. So the first is from a Bioconductor package called 10x Brain Data, which packages up a dataset released by the 10x Genomics company for single-cell RNA-seq. If that means nothing to you, that doesn't really matter. The point here is we have a matrix with about 30,000 rows and 1.3 million columns. If you were to store this in R as an ordinary array, you're looking at about 150 gigabytes just to hold the data. That's not great fun. From my own research, I work a lot on DNA methylation. We're part of the GTEx consortium, which you may have heard of if you're in genomics. For these data sets, we have the same problem in a different way. We have many rows, tens to hundreds of millions of rows, and tens to hundreds of columns. If we were to store these types of objects in memory, we're looking at nearly 0.5 terabyte in memory, which is more than the biggest node on our cluster at Johns Hopkins could handle. And that's before we even do anything with the data. So the pitch here is that DelayedArray is this one solution to this problem, and I think a very transferable one. So for instance, when we use DelayedArray with the 10x brain data, we have a summarised experiment that is only 180 meg in memory, and most of that is annotation about the samples, which we could store on disk, for instance, in a database, further reducing the size of the object. The methylation data, it's about 200 meg in memory. Most of that is information about the loci, the rows of this object. The nice thing about these having a small footprint is if you want to start an R session and load your object, it's near instantaneous. You don't have to wait for a serialized object that can take many, many minutes to load into memory. Now you might wonder how this is done. Well, it's basically cheating. You keep the data on disk. You don't bring it into memory until it's required. And specifically, in these two examples, we're keeping the data on disk in an HDF5 file. HDF5 is a hierarchical data format. It's a fairly broadly used scientific standard of sorts for numerical array data. And we wrap this HDF5 file within a delayed array. The nice things of this is we get a lot of the benefits that we had when we had ordinary arrays. It still feels and looks like an array, and that's through a clever design of the interface. So it has the same structure where rows are features, columns are samples. It has square bracket notation for subsetting that feels like an ordinary array. It has a matrix stats API that I developed called delayed matrix stats. So you can use all those tools that you were using for arrays now applied to these delayed arrays. You can integrate with matrix algebra with a little bit of extra work called block processing that I'll describe. It has a C and C ready sort of interface that I'll describe at the end of the talk. And I think the key thing here is now we've got something that is a small memory footprint that is conducive to interactive data analysis again. And I love doing my work that way, and so that's a real boon for me. So what exactly is it? Well, it's a few things. So it's a class. There is a DelayedArray class that is part of the DelayedArray package. But it's also a framework. It's available as part of Bioconductor, and as I say, it's developed largely by Hervé Pages. Like most of Bioconductor, this is using S4 object-oriented programming. You can install it. If you don't have Bioconductor installed, you can install the BioC manager package from CRAN, which is now the way to install Bioconductor packages, and then you can use that to install any package you want. So getting back to sort of the broad thesis of the talk and the abstract, I sort of conceptually think of a delayed array as similar to a tibble and dplyr. So from the delayed array description, we've got that wrapping an array-like object, which is typically on disk but needn't be, in a delayed array allows one to perform common operations without loading the object into memory. And in order to reduce performance would be a terrible idea. In order to reduce memory usage and optimize performance, we have these concepts of delayed operations and block processing. So from the tibble README, it's a modern reimagining of a data frame, keeping what has proven to be effective and throwing out what's not. And tibbles are data frames that are lazy and surly, getting back to the idea of sort of the analogy of delayed operations, where you don't do anything until you have to. A further bit is that, from the delayed array description, is that note that this also works on in-memory array-like data, such as capital M matrices from the matrix package, ordinary arrays, and that's similar to what dply's aim is, to abstract over how the data are actually stored, so that whether the data are local in-memory data frame or on a SQL database, you're writing the same code, and it kind of feels the same when you're doing an interactive analysis. Now you might be wondering, well, why delayed array? There are other options in R for disk-backed data structures. RHDF5 is the Bioconductor R package that interfaces with HDF5. There's also HDF5R. There are other things for disk-backed data like Matter, FF, Big Memory, or traditional database. My point here is that you can still use these. You can use a backend where it ultimately is just a big memory object, or it is an HDF5 file like what I'm using. I think the delayed array framework brings a powerful abstraction to these data where it is, like dplyr, it doesn't matter where the data actually are stored. You should be able to interact with it the same way. I think one benefit of delayed array, perhaps for Bioconductor developers, is that it's developed by the core team. So it is well written. I will say the documentation is not all there yet, and that's part of the reason I'm giving this talk, is to motivate some more documentation for it. But you'll have strong integration with existing infrastructure. So every delayed array has something called a seed, and that's where the data are actually stored. So the seed could be in memory, like an ordinary array, on disk, like I described with an HDF5 file, or even remotely served. So HDF5 have a Python HDF5 server software, so you can share your data, and it has an API where it's sort of JSON and requests, basically. So there's many options. To implement one of these backends, you only have to implement three things: a dim names, and a way to extract an array from this object. So to give some quick examples, Here's creating a delayed array where we're just storing using an ordinary matrix. Nothing too exciting here, just that you pass it as the seed of the object. And you see you get some nice printing where you don't have that thing where you have a matrix and it fills up your entire prompt. But we're not limited to ordinary arrays. We could, like I said, we could use a matrix, we could use a data frame, a tibble. We could use a slightly more complex object, which is a run-length encoded data structure, which actually works very well for the toy example I've given here and is quite useful for, say, coverage data in genomics. We can use an HDF5 file. That's implemented in the HDF5 array package. And we can have these remotely served ones that I described, which in this case is implemented in the RHDF5 client package, all available from Bioconductor. So every delayed array must have a seed, and the seed is closely tied to the concept of a backend. So there's these RLE array, HDF5 array, and RHDF5 client packages that provide these backends. It's user extensible. There's a vignette included in the delayed array for what you need to do to take an existing data structure and make it delayed array compatible. Good question is, what should I be using? I think right now, if you have data that is so large you'd need it on disk, the HDF5 array is probably the best choice. It may not remain that way, and I'm happy to discuss that in question time as to why I've arrived at that sort of opinion at this point. So I mentioned the concept of delayed operations. Here, we've got an example of a delayed array that has 3 dimensions, a very large third dimension with 90 million, whatever that third dimension is called, not rows and columns, but the third one. If we wanted to take that and add 1 to every entry in it, that basically returns instantaneously. If you compare that to, you see my mouse there? Great. If you compare that to if you had the object in memory, it takes about 7 seconds. The reason for that is it actually doesn't do the computation at this point in time. So just to give you a little explanation of that, there's this show tree function, which you can think of like the str function in base R, that gives you a little bit of information about the internals of the object. So when the object you start with has this sort of tree-like structure, when we add 1 to it, you can see we've added this line here that basically registers the fact that we've added 1 to this but hasn't actually done the operation at this point. If you take a subset of the data, it also is a delayed operation, but you can interact with it at that point. You can chain these, so you might take a subset and then take a transpose, and it will record those operations in a tree, and then when you want to sort of realize these operations, it steps through the tree and does the operations as needed. So that's the concept of realization. It's like you've done a whole bunch of operations on your array. You're like, okay, now I want to have the result because I need to do a plot or something. You can call the realize method. Generally, you don't actually have to do this, but it's in the background of a lot of the functions. You might realize it as a new HDF5 file, for instance. And this is just to illustrate that the object we started with, this is where it lives, and the realized version has been written to an auto-generated location. You can control that, but by default, it will write to an automatic location. You can also realize in memory. So you might have taken a subset and you're happy that, yep, I can bring that into memory, and you can do so. Block processing is the other main thing I want to get across, and that's like the issue of I have this large array and I need to traverse it to do some computations. These could be element-wise or block-wise operations. So for instance, we might want to go across the rows to compute the row sums. So we construct an array grid, which I've got the little code there for how you would do it in this particular example, and that will take rows of this object and step through. You might need columns. You can set up that for if you're doing column sums. You might set up a grid that is multiple columns. So you might know that, hey, I can bring in at least 5 columns worth of data, or up to 5 columns worth of data. That's going to be more efficient than reading in, it 5 times if you do it in one big hit. You can set up a grid that has that behavior. You might have one where it's a variable number of columns that you need to read in. For instance, if you were implementing the row sum functionality. You could go and say, well, I want the entire matrix. You probably don't want to do this. If you can read the entire matrix in, you probably didn't need to have it on disk to begin with. But that's the sort of the limit of this case. And then there's the idea of, well, there may be some optimal way to traverse this array. So if this is an HDF5 file, for instance, it stores the data in small sort of hypercubes on disk. So there is a way to extract what is the way that is most compatible with how the data is stored in its underlying data structure. So you construct these grids, and then in pseudocode you just iterate over these grids. So we loop over the grid, we read a block of the data into memory, and we apply our function to that block. The nice thing about this sort of structure is it's readily parallelizable. Here I'm using the BioC parallel package, and very little code has to change, and we can start processing these blocks in parallel. So if you have a machine with lots of cores, you get this consequent speed up. In practice, there's some functions to do this included in DelayedArray that abstract over some of these details. Some of these abstractions are still being worked out because sometimes you might have to iterate over multiple arrays. It's somewhat still being settled upon. And of course, you can write to blocks as well as reading from blocks. This is the one slide of the talk that is about something that I actually created. So this is a package, delayed matrix stats. The matrix stats package is something I really think is wonderful. If you're not familiar with it, it's very powerful. And I've just basically thought, well, I'd love to be able to call the same function regardless of whether I have an ordinary matrix or a delayed matrix. So my code is the same regardless. It's got complete coverage of the matrix stats API, some 74 methods, and it's implemented using these block processing techniques. It's continued development because sometimes we can come up with a more optimized method if we know something about how the data structure is stored. Finally, the beachmap package, which is developed by Aaron Lund with Hervé and Mike, provides a consistent C++ interface to matrices and delayed matrices. It uses RCPP and provides very basic methods to get and set rows and columns of this for implementing a C++ level functionality. So in summary, just the main point is that a delayed array is to an array as a tibble is to a data frame. It's a powerful abstraction for array-like data that may be in memory, it may be on disk or remotely served, and that you can use this framework directly, like we have in our methylation analyses, or you can extend it with other companion packages. And combining this with an on-disk backend, it's enabled analyses of datasets that would otherwise have been infeasible. So Hervé is the main person to thank for this work. Mike, Aaron, Samuela, and Chan, whose work I haven't talked about, have developed other companion packages. Martin Morgan, the head of Bioconductor, and my postdoc advisor, Caspar, for tolerating my explorations into this area and including it in part of his packages. There's some links. The first is to a actual biology paper where we use this to learn something about biology, studying DNA methylation in neuronal cell types. The beachmap package has a paper. In a couple of weeks, I'm giving a workshop on this at the Bioconductor meeting. The material will be available online. It's not yet, but it better be soon. And I did some pretty good URL sitting, so if you want to get the slides, it's all available there. Thanks. So the question was whether the alt-rep framework had been considered. At this point, no, it hasn't. I think there is good opportunities there, and it will be exciting. I think people are kind of waiting for the dust to settle a little bit to see how this is best done.
We're definitely talking to Hervé and Gabe, and people behind the alt-rep, people behind Delayed Matrix, definitely talking, and we're going to meet in September to pound out how we're going to do it.
Someone from Arcor in the audience is much better.